Yes, this is microbiology. Today I'm here to present the most important topic that is catalase test, and it is a biochemical test. So this is the main key point of our topic. Objective, we discuss objective of this catalase test and principle, procedure, and we discuss the QC. So this is the most important interpretation, result. Then we discuss application and summary of the catalase test. Objective. The main objective of this test to find gram-positive cocci such as Streptococcus bacteria and Staphylococcus bacteria. These two bacteria, they are uh, and they are category under classification of gram-positive cocci. So Staphylococcus bacteria having enzyme catalase, so they called catalase positive, whereas Streptococcus bacteria have not, so this is called catalase negative. The test actually help in diagnosis of these two cocci. Principle. The principle of the catalase test is very important. Everybody should know about the basic principle of the catalase test. So the enzyme catalase mediates the breakdown of hydrogen peroxide into oxygen and water. The presence of enzyme and bacterial isolate is evident when a small inoculum is introduced into hydrogen peroxide and the rapid elaboration of oxygen bubble occur. The lack of catalase is evident by lack of or weak bubble production. So basically we know the Staphylococcus bacteria, they having enzyme catalase. If they are catalase positive, so this enzyme broken down the hydrogen peroxide into oxygen and water, and as a result, we have a bubble in a medium. So if there is no bubble found in a medium, so we say these are catalase negative and it will be a streptococcus bacteria. So the main point we should know is that the culture should not be more than 24-hour old, because if the culture is older than 24 hour, so the enzyme, we already know the enzyme, they are not stable for a longer period, so the result will be falsely negative or falsely positive. And so why the bacteria that is broken the hydrogen peroxide into H2O2 and oxygen, because it's protect themselves from the toxic effect of hydrogen peroxide. So actually it helped in the defense of the catalase positive bacteria, the bacteria which are catalase positive. So basically, catalase enzyme show depends against the hydrogen peroxide. So here is the diagrammatic principle of catalase test. When catalase positive bacteria present, so catalase broken down this hydrogen peroxide into H2O2 to water molecule plus oxygen. So as a result, gas bubble will produce. Procedure. Uh, within the procedure we have two methods. First one is tube method and the second one is slide method. The tube method, within the tube method we take 2 ml hydrogen peroxide solution, and then using a sterile wooden stick or glass rod takes several colonies of 18 to 24 hour test organism and immersed in the hydrogen peroxide solution. Observe for immediate bubble. So we must know the colonies that we pick up from 24-hour organism. 24-hour. So if the colony is more than 24-hour, if the colonies we take, if the colony of the organism we take from more than 24 hours, so the result will be falsely negative because the enzyme catalase is not stable for a prolonged time. So if there is a bubble produced, so it's mean they are catalase positive, and it will be staphylococcus. Slide method: use a sterile wide loop for transfer small amount of colony on the surface of clean glass slide, which will be dry, and place a drop of 3% H2O2 in the glass slide. Then observe for the evolution of oxygen bubbles. If there will be appearance of oxygen bubbles, it means they are catalase positive. If there will be absence of oxygen bubble, it's mean they are catalase negative. So here you can see a two image, slide method and tube method. Catalase, when catalase positive, there will be of oxygen bubble production, and when the catalase negative, there will be no production of oxygen bubble. And the tube method, we have a catalase negative, we have no bubble in the tube, and catalase positive, we have see of bubbles in the tube. The one thing should be remember, we take a cartoonist from 24-hour organism, not more than 24 hour. So if we take more than 24-hour organism sample, the result will be falsely negative. So quality control must be performed on every single test, either they are in other departments. So QC of the catalase test is very important for the to assure the accuracy of the test. So within the catalase test, we have positive control, and another one is negative control. Positive control is Staphylococcus aureus ATCC 33592, and negative Enterococcus faecalis ATCC 29212. These two controls should be run along with the test. If we perform catalase test, so we must be run these two control. So the basic summary of catalase test is that the test is basically help in diagnosis of staphylococcus. So because staphylococcus bacteria are catalase positive, whereas streptococcus are catalase negative, so it help in the diagnosis of staphylococcus bacteria. Thank you for watching.